नमस्कार मित्रांनो, टेक मराठी या यूट्यूब चॅनलवर तुमचे स्वागत आहे. मागच्या व्हिडिओमध्ये आपण बायोलॉजी प्रॅक्टिकल एक्झाममध्ये येणारे एक्सपेरिमेंट कसे लिहायचे ते पाहिले. तो व्हिडिओ तुम्ही पाहिला नसेल तर नक्की पहा. लिंक तुम्हाला डिस्क्रिप्शनमध्ये दिलेली आहे. या व्हिडिओमध्ये आपण पार्ट बी स्पॉटिंग कसं लिहायचं ते पाहणार आहोत. प्रत्येक स्पॉटला किमान 4 ते 5 पॉईंट लिहिणे आवश्यक आहे. चला तर मग व्हिडिओला सुरुवात करूया. 1 बाय 1 बायोलॉजी प्रॅक्टिकल बुकमध्ये दिलेल्या क्वेश्चन पेपरप्रमाणे सर्व स्पॉट पाहू. स्पॉट ए मधील पहिला स्पॉट पाहू. वाइंड पॉलिनेशन. द गिवन प्लांट स्पेसिमेन शोज वाइंड पॉलिनेशन anemophily by maize flower. The flowers are unisexual, small, non-attractive. Flowers are without color, odor, and nectar. Anthers are versatile, which produce large number of tiny, lightweight, dried rust, like pollen grains with smooth enzyme. Gynoecium shows feathery or brush-like stigma with long style. Style and stigma are bushy. Flowers are unisexual and protandrous, so it is cross-pollinated by wind. Stigma is feathery to trap pollen carried by wind currents. 2. Insect pollination. The given plant specimen shows insect pollination, entomophily, by salvia flower. Flowers are bisexual, attractive, and have bright colored petals. The nectar and nectar glands present in flower. Salvia shows bilipid corolla. The larger lip encloses stigma and style. 2 stamens located at mouth of corolla tube. The flowers produce sweet odor smell. 3. Emasculation. The given step of hybridization technique is emasculation. Removal of young anthers of the stigma before anthesis. Formation of pollen grains. The bisexual flower of a female parent is emasculated. Buds are open and stamens are removed mechanically by forceps or by hot water treatment. Emasculation done to avoid self-pollination. Emasculation is done in the early hours of morning. Date and time of emasculation is recorded. 4. Bagging and tagging. The given step of hybridization technique is bagging and tagging. Emasculated flowers are bagged in polythene bags or sterile paper bags. It prevents pollination by foreign pollens. This is called bagging. A tag is tied to the bag. The tag carried brief information about the name of selected parent varieties and the day, date, and time of anthesis, emasculation, and crossing. आता आपण स्पॉट बी मधील पहिला स्पॉट पाहू. टू स्टडी रेट ऑफ ट्रान्सपिरेशन. The given experiment setup is to study rate of transpiration by 4-leaf experiment. Take 4 leaf and apply petroleum jelly on surface of leaf. Label leaf as A, B, C, D and applied jelly on the both surface. On lower surface, only on upper surface, not applied. Observation shows first leaf remain fresh, second leaf slightly dried, third more dried, and fourth leaf completely dried. So rate of transpiration is higher on lower surface of the leaf than from the upper surface. The given experimental setup is to study separation of plant pigments by paper chromatography. 1 On the strip, we can observe the separation of photosynthetic pigments as bands of different colors in particular sequence. From the loaded spot up to the top, the sequence is chlorophyll B, yellowish green, chlorophyll A, bluish green, xanthophyll yellow, carotene orange. Spot C, first Calotropis procera, Rui. The given xeropathic plant is Calotropis procera, Rui. It is non-succulent, wild shrub of desert and wasteland. The leaves and young branches are covered by a mainly coating along with hair which act as insulating covering. The leaves are thick and somewhat leathery. The plant process latex. Second, Acacia arabica, Babul. The given xeropathic plant is Acacia arabica. It is non-succulent, xeropathic. The leaves are bipinnately compound. Leaflets are very small in size to reduce transpiration. The stipules are modified into spines to reduce transpiration and also to protect plants against grazing animals. The older parts of stem are covered over by thick brown bark. 3. Opuntina dilleni. The given xeropathic plant is Opuntina dilleni nakfani. It is a succulent xerophyte. The stem is flattened, jointed, and green. The stem is fleshy due to presence of mucilage that retains water. Leaves are modified into spines. 4. Acornia. The given aquatic plant is Acornia water hyacinth. It is free-floating hydroid that grows in freshwater ponds, lakes, etc. The stem is short and spongy due to parenchyma tissue. Leaves show swollen spongy petioles and arise in cluster at node. Adventitious roots are present which act as a balancer. 5th, Typha cattail. The given aquatic plant is Typha cattail. It is an amphibious and anchored hydroid. The plant grows in marshy places or shallow waters. The leaves are long, linear, soft, spongy, thick and subcylindrical. The leaves show the presence of mechanical tissue. Spot D, TS of testes. The given permanent slide is TS of testes. The testis is externally covered by fibrous connection tissue called tunica albuginea. It is internally covered by tunica vascularis and externally by incomplete covering tunica vaginalis. It is externally covered by incomplete covering tunica vaginalis. Few large pyramidal cells are present called nerve cell or Sertoli cells. Sertoli cells provide nourishment to sperm till maturation. Second, TS of ori. The given permanent slide is TS of ori. It show outer cortex and inner medulla. The cortex is lined by germinal epithelium. The medulla shows connective tissue, stroma. Cortical regions show different stages of developing ovarian follicles as primordial follicle, primary follicle, secondary and graphene follicle. Third, VS of blastula. The given permanent slide is VS of blastula. It shows outer layer of small flattened cell called tropoblast. It encloses a cavity called blastocoli and inner cell mass. The inner cell mass attached to one side of trophoblast. The trophoblast cell is contact with the inner mass are called cell of rubber. Spot E. Turner syndrome. The given karyotype shows disorder Turner syndrome. Individual born with Turner syndrome has 44 autosomes and with only one XX chromosome. Hence, it is also named as X monosomy. They have a short stature and webbed neck, lower posterior hairline, broad shield-shaped chest, poorly developed ovaries and breast, and low intelligence. They are phenotypically female. Second, Klinefelter syndrome. The given karyotype shows disorder Klinefelter syndrome. It is chromosomal disorder caused due to extra X chromosome in males. This genotype of an individual is 44 plus XXY. Extra chromosomes is a result of non-disjunction of X chromosome during meiosis. They are described as a feminized males and has masculine development. Voice pitch is harsh and have underdeveloped testes. They are tall with long arms, feminine development and no spermatogenesis. Therefore, individuals are sterile. Down syndrome. Third, the given karyotype shows Down syndrome. This syndrome is caused due to an extra copy of chromosome number 21st. It shows presence of 3 copies of 21st chromosome instead of homologous pair. These patients have mild or moderate mental retardation. Skeleton development is poor. Face is typically flat and rounded with open mouth. Spot F. Disease, malaria, control, proper arrangement to prevent the mosquitoes from entering the home. Prevention of mosquito bite by using mosquito nets. Spraying insecticide and draining stagnant water where mosquito lays eggs. Mosquito larvae can be controlled by using gambusia fish. Cleanliness, spraying of insecticides to kill mosquitoes. Control measure: wash hands with hot water and soap after using toilet and changing baby's diaper. Drink boiled water or water must chlorinated and filtered. Avoid eating unhygienic food. Vegetable must be properly washed and cooked. Proper sanitary facilities including sewage disposal help in prevention. Third, disease ascariasis. Control measure: avoiding defecation in open space, prevent the spread of ascariasis. Personal hygienic habits like washing hand with water and soap after using toilet and before eating, washing vegetables before cooking, avoiding raw vegetables is important for prevention of ascariasis. 4. Disease ringworm. Control measures: keep your skin clean and dry, avoid close contact, sharing of clothes, sport equipments, washing clothes in hot water with fungicidal soap. Spot G. Stages of meiosis. First, metaphase 1. The given permanent slide shows stage of meiosis. Metaphase 1. An imaginary line develops at the center of the cell. Bivalent are arranged at the equator of the cell. At the end, spindle formation take place. It helps in arranged and movements of chromosomes. Members of each bivalent are connected to only one of the two poles, but opposite poles. Second, anaphase 2. The given permanent slide shows stage of meiosis. Anaphase 2. Centromere divides and sister chromatids get separated, which are now called daughter chromosomes. Spindle fibers start considering becoming shorter and shorter, pulling daughter chromosome up to the opposite poles. Third, telophase second. The given permanent slide shows stage of meiosis. Telophase second. Daughter chromosome get collected at the opposite poles. Reappearance of a nucleolus and nuclear membrane result in the formation of two daughter nuclei. Spot H, xeric animal. First, camel. The given spot of a xeric animal, camel. It is Zirco's animal adapted to the desert condition. It excretes concentrated urine in order to conserve water. It can tolerate wide range of temperature fluctuations. Camel can even close its nostril to stop blowing sand from entering nostril. Flat and wide feet help it to walk easily over soft sand. It is capable of drinking large volume of water, about 100 liters. It is a short time, about 10 minutes. It has long eyelashes that protect eye from sand strong winds. Second, kangaroo rat. The given spot is of xeric animal, kangaroo rat. It is a Zirco's radiant. It is nocturnal in habitat. It seals its burrow during daytime to prevent the loss of moisture. It obtains metabolic water. Kangaroo rat neither sweat nor pant to keep itself cool. Aquatic animal. The given aquatic animals, rohu. The given spot is of aquatic fish, rohu. Body is laterally compressed and streamlined. It shows presence of gills for respiration, which help in exchange of gases in water. Paired fins help in swimming, and caudal fin act as a steering during swimming. Body is covered by scales to prevent osmotic entry of water into the body. 4. Animal, dolphin. The given spot is of aquatic animal, dolphin. It is mammal, not a fish. It has streamlined body, smooth and fearless body to reduce friction. It has a backward covering dorsal side and dorsal fin. The position of nostril near the top of the head facilitates easy breathing when animal reaches the surface of water for breathing. Flippers in dolphin can control for steering, slowing and going up or down or out of water. Spot I. The given spot is Vs of anatropous ovule, funiculus. It gives support to the body of ovule. It supplies food and water to developing ovule. Nucellus. It produces female gametophyte at maturity. It is nutritive function. Integumates. that gives protection to nucellus and embryo sac microphyle. It allows the pollen tube to enter the ovule during the process of fertilization. Embryo sac. Embryo develops in embryo sac after fertilization. Chalaza. It produces integument's from the basal part. Egg. After fertilization, it converts into diploid zygote, which by mitosis produce an embryo of seed. Synergids. One of the synergid help to give entry for the male gametes within embryo sac. Antibodies. After fertilization, the degenerates and act as nutritive tissue. Secondary nucleus. After fertilization, the diploid secondary nucleus convert into a diploid primary endosperm nucleus, which produce triploid endosperm. Second, functional areas of cerebrum. The given model of brain, it shows functional area of cerebrum, frontal lobes, motor area, controls voluntary motor activities, premotor area, center for involuntary movements and autonomous nervous system, association area, coordination between sensation and movements, Broca area, it translates thought into speech. Spot J, rolling of tongue. The given pedigree chart shows genetic trait rolling of tongue. The rolling of tongue is the ability of a person to roll the tongue in U shape. It is caused by autosomal dominant allele. Inability to roll the tongue is caused due to autosomal recessive allele A. Both homozygous dominants AA and heterozygous AA individuals are able to roll the tongue, while homozygous recessive allele AA individuals are unable to roll the tongue. Widow's peak. Second, the given pedigree chart show genetic traits widow's peak. Widow's peak is a V-shaped hairline across the forehead. It is dominant autosomal trait. The gene responsible for widow's peak is dominant W. Both homozygous dominant WW and heterozygous WW individuals have widow's peak. Homozygous recessive WW individuals have straight hairline. This feature is observed with both man and woman. Third, the given pedigree chart shows genetic trait ABO blood group system. The blood group in human being are described as per ABO system of classification. The gene I control the ABO blood groups. It has the three alleles IA, IB and small I recessive. Each individual possesses only two alleles out of three. Alleles IA and IB are codominant. Individually, they are completely dominant over allele I. Fourth, color blindness. The given pedigree chart shows genetic trait color blindness. Color blindness is a sex X-linked recessive disorder of humans due to recessive gene present on X chromosome. Color sensitive cone cells are not formed. This results in red-green color blindness. It is more common in male than female. It follows crisscross inheritance, as this is transmitted from the father to the grandson through his carrier daughter. Thank you.